This tutorial inspects the differential expression results reported by the application once intermediate quality steps such as filtering, flagging, or CT normalization have been performed. First of all, let us have a look at the settings that we can configure using the project options. This is the most important information that StatMiner needs to automatically perform a differential expression analysis. We will later see how to define other parameters such as the scale of the RQ reports. So we have three different statistical tests to evaluate whether two groups are significantly different or not. It means to check if there is any kind of regulation between the target and control group. Lemma is an improved version of the parametric student t-test. This test is recommended when our data follows a normal distribution. If apart from the conditions defined by our experimental factor, there exists an alternative factor that links our samples within biological groups, for instance, the time on a treatment analysis, then there is a chance to include this information of our linked samples in the statistical test calculation. This can be done by selecting the secondary factor from the experiment design that we have previously defined. Finally, and in case we are not sure about the shape of our data distribution, it is recommended to use the non-parametric test, Wilcoxon. As we will see later, those statistical tests do not have any influence in the RQ calculations, but just on the p-value that will decide whether certain detector is regulated when it is compared with the control group. Only one more step is needed before running our analysis, and that is to define our main experimental factor in the control group. The automatic analysis will compare all the experimental conditions with selected control by default. Once the analysis is done, we will have the opportunity to iterate over the expression analysis by performing one-on-one comparisons, for instance, amygdala group versus selected calibrator. We are now ready to go. StatMiner is automatically preparing our data before giving an initial report of expression. As mentioned, the first report that we can inspect is the RQ value of every detector along all conditions versus the calibrator. For now, we will focus on one-on-one comparison. In this example, we have three different tissues plus the control group. We are going to compare amygdala tissue versus control to get top-regulated detectors and later check how those detectors behave in the remaining tissues, namely cerebellum and cortex. Before running the RQ calculation, we have the option to select the FDR method. This adjustment of the p-value is highly recommended when we are performing not one but multiple statistical tests, as is the case when we are evaluating every detector. In this case, we have more than 300 contrasts. We can also set the alpha significance level. All detectors with a p-value less than this threshold will be considered regulated. And finally, the RQ scale. In this case, we can keep a logarithmic scale selected in order to better inspect the RQ values. So, here we go. This is the relative quantification report for the amygdala group versus control. From left to right, we have a data table summarizing all the information calculated: delta CT, raw data, quality values, RQs, mean and deviation by groups, etc. Delta CT profiles for the groups that we selected for the comparison. All reports will be colored by the amplification flags to alert poorly amplified detectors. RQ bar chart has been split into non-significant and significant detectors using the logarithmic scale. And finally, we have the volcano plot representing the RQs versus the adjusted p-values. Our goal is to retrieve the top regulated detectors, so let us start our inspection from this volcano chart. First step is to clean the report from detectors with very low levels of amplification, since we cannot rely on them. Next is to focus on detectors with troubles in the calibrator group, target, and finally amplification issues in both groups. Now it is time to filter out non-significant detectors, meaning the detectors that according to the statistical test display no significant differences between the expression of both groups. We have now filtered for regulated detectors, but would like to identify most up or down regulated detectors in this dataset. To do that, we can use the RQ filter component by inverting the selection. This way we can keep values from both sides. Here we see the most downregulated detectors, and over here the top regulated ones. What we are going to do now is to use the Detector List Manager tool to save them into two separate lists. This will allow us to interact later with other reports in the application. To do that, we can select the top regulated genes and create a new list with this selection. This is the upregulated detectors list. And now we proceed in a similar way by selecting the most downregulated detectors. And here is the downregulated list. Now, let us go back to the rest of the report, but before that, let us close all the filtering components. As we can see, the expression reports are synchronized so that they display exactly what has been kept in the others. Now that the profile has been properly filtered, it is easy to identify and validate expression results. These are the top regulated detectors, and as expected, delta CT values present a very low variability within these groups. Statistical test rejects high variabilities. And there is a large difference between the means of the groups. Let us go back to the all versus control report to check what happens with our detectors of interest when compared with the rest of the groups. We can now focus on inspecting the most regulated detectors, and we can use the zooming tool to verify that our detectors have a similar behavior along tissues, with maybe some exceptions in the cerebellum tissue. This is how resetting the zoom action works, and we are now resetting the filters. It is important to know that we can send these data and visualizations to a global report that can later be exported to formats such as Excel, PowerPoint, and PDF, etc. Please watch our subsequent tutorials to get a deeper understanding of StatMiner.